visits and expensive medications. I've seen this firsthand. Part of my postdoctoral work involves clinic visits. So I'm actually watching people that have lost their limbs undergo wound debridement where affected tissue is removed. It's not pretty. So I focus on why these wounds are chronically inflamed and how we can promote healing. The stages of healing and repair are divided into inflammation, which is the initial response that mediates tissue repair. I just showed a little schematic here with immune cells flooding in from the bloodstream, initiating the inflammatory response, followed by proliferation, where your damaged blood vessels begin to grow, creating a supply of cells and nutrients to that wounded repair tissue. And you also have that nicely formed granulation tissue, which is fancy word for newly formed tissue. And inflammation calms down, finally leading to the remodeling stage where the wound has reformed, the tissue architecture has reformed, the wound has closed. With diabetic foot ulcers, proliferation remodeling doesn't happen. Think of like potholes in concrete. That pothole never reforms. And if you live in Connecticut, you know how much damage it can do to your car. So taking a closer look at what this actually means when I say these wounds are stuck in the inflammatory stage, there's persistent inflammation in the wound tissue. So again, that redness, that open sore. The later modeling or no remodeling prevents rebuilding of the tissue architecture. And open wounds, again, remain susceptible to infection or damage, which can lead to amputation in most serious cases. So my work focuses on using diabetic foot ulcers and non-diabetic foot ulcers, trying to understand which biomarkers we can use to develop better treatments. I use single-cell sequencing, histology, and mRNA expression. I'll just show a few bits of data here. Single-cell sequencing of diabetic and non-diabetic foot wound tissue will show you differences in the tissue architecture. Most notably, we see decreased keratinocytes, which are important for skin rebuilding. But interestingly, we see more fibroblasts, yet remodeling is delayed, so what are they doing? And also, about the same number of macrophages, yet diabetic tissue is supposed to be more inflamed. You'd expect macrophage numbers to be much higher. So thinking about the other techniques I'm using to interrogate this tissue, I turn to histology. I've shown here non-diabetic foot wound tissue and diabetic foot ulcer tissue. You'll notice that CD45, really beautiful green stain, CD68 positive, macrophages in red, whereas in the foot ulcer tissue, we see hardly any CD45, but we do see spots of macrophages. So this proposes a lot of questions about what's happening with the immune system. And those of us that do human work, I'm sure, are very fond of IRBs and patient recruitments and trying to get that N number high, which is always a challenge. So in thinking about how I can interrogate this immune response, I have to think about other models I can use. My work uses mice, specifically diabetic mice, trying to understand how this dysregulated immune response is promoting this pro-inflammatory chronic wound. And more specifically, how can I modulate this behavior? Using a mouse, I didn't show here, I wasn't sure how people would respond to seeing mouse wounds, but on the backs of these mice, I use a 4mm punch biopsy tool to create even wounds, 6 wounds on the backs of mice, and I'm able to collect wounds on days corresponding to the stages of repair I mentioned previously and interrogate them further. But as I mentioned in the clinic, these visits are very intense and some people end up losing their limbs. They do everything the doctor tells them to. They change their diet, they exercise, they haven't had ice cream in 20 years, but it's not working. So if diabetes is so destructive, where there are people on the outliers that are not responding well to treatment, these wounds aren't healing, they're suffering even more, it's forcing me to think outside the box, or I guess in this case, outside the body. So what tools can I use to challenge the destructive nature of diabetes? So I also incorporate biomaterials as a means for intervention to improve wound healing. Biomaterials are engineered substances that interact with our living systems. And specifically, I use hydrogels. These are water-based materials, think of like hair gel or like Eco-Styler, and they retain their structure over time, and they act as the concrete to fill in that pothole of the wound. Hydrogels, in general, can deliver anti-inflammatory properties, specifically with the diabetic wound, and can promote healing and tissue remodeling. So early stage intervention in my mice has shown that repeat administration has improved wound healing And we've created this barrier between the outside world and the wound environment that allows that or prevents repeat damage. So thinking about how this will help people, and if you're interested, stay tuned. Humble plug there. What I envision is these wounds will be able to be sealed, not in a painful way that will prevent people from having to come back to the clinic to undergo that debridement, but creating a space, creating a material that will harden on the bottom of their feet, preventing that tissue from enduring more damage. The tissue responds really well to the hydrogel, and so there is no issue of it popping off. And eventually, we would have a healed wound response. So my greater vision for this work is much like what happened with the COVID tests, how we were able to get them delivered to our home. Being in the wound clinic, I'm coming face-to-face with people that have very serious challenges and barriers with the medical system. Getting to the clinic, getting appointments, access to healthcare, access to a CVS that carries their drugs. My hope is that I'm able to not only create a gel that works in mice and creates a really cool paper, but I can create an at-home kit that people can administer themselves in the safety and comfort of their own home. All of this work is the foundation of what I hope to be, or no, not hope, what will be the Carter Lab. So taking clinical research, interrogating the immunology of this tissue, and considering diseases that are destructive, like diabetes and cancer, using biomaterials to then alter that immune response, promoting healthy, healed responses, and then giving it back to the community. So using mouse models, and then hopefully my kit, with the overarching aim of using tissue engineering approaches to promote regeneration of disease and damaged tissues, not only to promote repair, but creating accessible treatment options. And hopefully one day you'll see me on Shark Tank peddling my hydrogel kit. And so with that, I'd like to thank my PhD group, tendon biology group led by Neil Miller, my postdoctoral group led by Valerie Horsley, countless friends, family, and supporters, and especially a few groups I want to highlight is the Yale Black Postdoc Association, the National Black Postdoc Association. Postdoc life is hard, isolating. Without them, I would not be here. And so with that, I will take any questions.
Great talk. Am I going? All right. So I have one naive question. How does that hydrogel stick to the skin? And then my second question is, have you played with the stiffness of the materials? Because, for example, there is some evidence that change in stiffness in these biomaterials can promote or inhibit regeneration in different contexts.
So I'll answer the second question first. So there's actually a postdoc in the Horsley lab. She works on biomechanics, and she's looking at fibroblast response to stiff and soft materials. So that is something that's definitely coming. With the hydrogel, it's thermoresponsive. So at 4 degrees, it's really nice and liquid, and once it touches a warm surface, it adheres. And then over time, it degrades. I always say macrophages are like the toddlers of the immune system. If they're not happy, nobody is. You'll see in my work that macrophages respond really well to the hydrogel. They degrade it very nicely. And after about 20 days, it has fully been incorporated into the mouse wounds. But stay tuned.
Hi. So Jay, for those that can't see me, at Fox Chase Cancer Center. So you mentioned fibroblasts, and you mentioned that there are higher fibroblasts in the diabetic ulcers. And you said, what are they doing there? And I don't know if that was bait, but you baited me. So what do you think they're doing if anyone has started to look into that? And then if not, maybe we should collaborate when the future Gardner and Carter labs exist.
Well, one, yes to collaboration. Always good to make friends. As far as this fibroblast population, it's pretty strange. I honestly think this sample is a bit of an anomaly. Looking at my other histology, looking at alpha SMA staining, you don't see this increase in alpha SMA in my diabetic foot ulcers. So this may have just been the sample that we chose. They had higher fibroblasts, and maybe they are being produced more to try and start the remodeling process. But I guess, okay, so maybe if we have chronic inflammation in the wound, maybe those fibroblasts are persistently being activated by macrophages, for example. You have a, excess inflammatory response, excess fibroblasts, differentiation to myofibroblasts, and that could be what we're seeing. But they could be dysfunctional, which is why we don't see that increase in matrix protein expression. We don't see that filling in of the cavity. But collaboration on the table.
Yeah, definitely the collaboration.
I'm in, baby. I'm in.
Yeah, because so just for a note though, at least for looking at alpha smooth muscle actin as a marker, we've seen sometimes that like fibroblasts can almost reach an exhaustive state where they kind of lose alpha SMOS signaling, but they're still active. So that may not be the best marker. Right now, we've kind of been using potoplanin as a really good fibroblast marker that seems to be panning out. That maybe that could be something that you look into to see if there's enough fibroblasts actually in your samples. Yeah,
so I've been using, going off on a tangent now. We could talk later too. I've been using potoplanin as a stromal cell marker, potoplanin and CD90. I was thinking of using FAP, fibroblast activating protein, because a lot of head shakes. Okay, no.
Both me and Rebecca like, nah.
We'll talk later. Thank you for your question. Glad I asked for the order of the antibody.
Yeah. Okay. Yes. Yeah, I won't ask about fibroblast. I had two kind of so do the macro? I don't know how you would be able to know this, but are there more? Is the lack of immune cells in the wound, like if you could somehow get an unwounded diabetic foot, would you have fewer immune cells there than an unwounded non-diabetic foot? Like, is the starting state just different, or is it also the wound response is different, or like both? I don't know if you can do that experiment. It seems, now that I say it out loud, a little challenging.
Actually, we did flow cytometry, looking at the early stages of wound healing, and in the mice, obviously, because the foot tissue
the mouse is a nice model for that.
Right, it's good. So the initial stages of wound healing, the macrophage influx is the same, but it just persists. So I would look at like day 0 to day 3 in control and diabetic. The numbers are the same, quantification of neutrophils and macrophages, which is interesting because you think, and conceptualizing this, I thought, okay, vascular dysfunction, crappy blood vessels, whatever, maybe they aren't able to get to the wound site, but they're actually there.
They're just not doing the right thing.
Yeah, so I'm like, what are they doing? And how can I reprogram them? So, yes.
Yeah, so that gets my other question of have you, now I'm more of a biomaterial nerd. Can you add things to your hydrogel and functionalize them to potentially get your cells to behave? All right. Okay.
Next. No, but excellent question. Yeah. Stay tuned. Something I'm working on.
Me? Yeah. Okay.
Hi. Keelan, Sylvester Comprehensive Cancer Center. Okay, 2 questions. So the first one is, I know you said you do patient clinic visits. I have to ask I'm not familiar at all. At the population level, are there differences in the number, are there differences at the population level in the amount of ulcers that form in diabetic patients, and the severity of those ulcers? And then the second separate but related question is, when you think about the fact that inflammation is a mediator of stress and metabolic disease, to what extent can you account for other types of psychosocial stress that might affect an immune response? And have you thought about how that might affect the way that your intervention actually works across different types of folks?
Yeah, amazing loaded questions. I love that. The first one you asked about location and quantity of wounds. So some of the diabetic patients that come into the clinic are wheelchair bound, and some of them have most of their wounds on the backs of their thighs and their legs. So it's not, it usually starts off as a foot, on a toe, but then, the infection is too bad and they can't walk, but then they end up developing more. So yes, that does happen. And then you do have some that diabetics that come into the clinic with starting off as a toe wound, they end up cutting off the toes. Cutting off the toes. It's okay. And then they end up getting more wounds around their ankles. So the numbers do differ between those who are non-diabetic and diabetic, but also mobility challenges as well are playing a role. As far as other stressors, thinking about the barriers of like the healthcare system, there was a woman who came in, she was really stressed. She couldn't get an Uber and she was late and missed her appointment and they wouldn't reschedule her. And it's hard being in the clinic, but it's, part of it. And so thinking about the stress that she's undergoing and she's limping, trying to like rush in the parking lot and like, that's hurting her wound. Like that's not good for her. And so if there was a way to say, we will call a cab for you, you come comfortably, we'll give you a walker, we'll put you in a chair, something like even just getting her to the clinic, to the actual clinic room could reduce any kind of further damage she could do just in that situation. So thinking about lifestyle as well could definitely, absolutely play a role in the biology that we're seeing. So thank you.
Yeah, thank you, Dr. Carter.
Thanks.
Moving on, I'd like to introduce our second speaker for this session, Dr. Gina Gallego, who received her PhD from Washington State University in Immunology and Infectious Disease and is currently doing a postdoc within the Morgridge Institute for Research and the University of Wisconsin-Madison in the Department of Microbiology and Immunology.
Hello everybody, can you hear me well? Good. So I'm happy to be here to share with you the research vision of my future lab, that is understanding the metabolic changes induced in host cell by enteric apicomplexa parasites. But first, I need to introduce you what are these apicomplexa parasites. So these parasites are characterized by the presence of three organelles. They are rhoptia, micronems, and spherical bodies that sometimes they are called dense granules. Between this important phylum of parasite, we have different microorganisms. We have Toxoplasma, we have Cryptosporidium, Plasmodium that cause malaria, we have Theileria and Babesia that affect mainly animals, and all of them have very important, produce a lot of important morbidity and mortality in humans and animals. So normally in a parasitology talk, people present the life cycle of the parasites. But today I'm going to present my research life cycle. So I started this research journey early back in Colombia doing my undergraduate studies. I did my bachelor degree in La Universidad del Tolima, and I did internship. I worked for the immunology institution in Colombia, working in Plasmodium. I participate in projects in vaccine development, and then I also learned a lot of techniques in immunochemistry. Then I got my scholarship to move here to the United States. I did my PhD in Washington State University, where I studied Babesia. I learned a lot of molecular biology techniques and proteomics, and then I moved to Wisconsin. And in Wisconsin, I started working with Dr. Laurel Knoll, who is a famous parasitologist who studied Toxoplasma. But also, I get a multidisciplinary fellowship that have 2 PIs. So my second PI is Dr. Melissa Scala, who is an engineering and work more in cancer and developing tools to study cancer. So I'm the perfect mixture of both of them. So I work in parasitology and applying these engineering techniques. And I'm mainly in my postdoctoral studies studying Toxoplasma and Cryptosporidium, but in my future lab, I want to focus more on Cryptosporidium. So why these parasites are too important? So all of these parasites produce a lot of outbreaks around the world. Here I have some examples. So in Wisconsin, where I am located, in 1993 was a very important outbreak, maybe some of you remember, where people have to boil water even to brush their teeth. So in that outbreak, more than 400,000 people was affected. And here we have other example why these parasites are important. So as we can see, affect different parts of the water, are mainly transmitted by water, but also by food, affect mainly immunocompromised patients and children under 5 years old. Also here, other example, toxoplasmosis also produce a lot of outbreaks. Here, as we can see in this map, and also is transmitted by meat, vegetable, water, and milk. So why is important to study these metabolic changes inducing the host cell by these parasites? So it's very important because the parasite is a parasite, so it depends on the host to be able to replicate and produce a lot of metabolites. For example, the parasite cannot produce the purine rings, so they need to steal from the host. Also, it's important because the metabolism of the parasites behaves sometimes similar to cancer cells. Also, it's important because many of these metabolites could be very important drug targets. So now, this is my research vision of my lab, and I want to study, as I mentioned at the beginning, toxoplasma and also cryptosporidium. These are dysenteric apicomplexa parasites, and what really interests me is that there is a lot of data, or some preliminary data, that said that the infection with these parasites are a risk factor to develop colon cancer. So I want to try to understand how from chronic disease we can move to cancer, but I need to take little steps. So I have 3 aims that is going to allow me to know better the metabolism of these infected cells, and I will describe each of these aims. So first, I want to study mainly redox biology. In the second aim, I want to study more host nucleotidases, and in the third aim, I want to study more glycolytic demands. So let me start with the first one. That is why to study, why to understand the redox biology in the infected host cells. So this is important because there are many unknown mechanisms in redox biology. This redox biology influences immune response, and again, is a very important target for drugs development. So I use optical metabolic imaging technique, that is an engineering technique, to study these parasites. So using this technique, we are able to measure NADH and FAD in a non-invasive way in real or physiological conditions. So we are able also to calculate optical redox ratio and all these parameters that we can see here in this table. So using optical metabolic imaging in Toxoplasma gondii infection, with the same field of cell, we can have measure of NADPH, FAD. I use a Cheruiyot parasite to be able to distinguish between uninfected and infected cells. So using this approach, we found very important changes at the level of NADPH during 48 hours post-infection. These are infected cells, and in color we can see the fluctuations in NADPH. Also, we found fluctuation in FAD. So using other techniques, sometimes it's not very easy to have these measures. So I'm very happy using the optical metabolic imaging for that reason. Also, we found that the infected cells over time trying to become more oxidized. So here we see the same result by using quantitative way. We compare with the uninfected cell, and we see that the infected cell have a more oxidized redox ratio. So, but there are still many questions to answer. So in my future lab, I want to study Toxoplasma gondii infection in different organoids. We have in the lab growing placenta organoids, regna organoids, intestinal organoids. In Cryptosporidium, I want to study also in different colon cancer cells. And we want to study how the different stage of the parasite change the redox biology of these infected host cells. Also to test antiparasitic compounds and to check how they change the redox biology of the cells. Study the gene expression. For example, in Toxoplasma, there are at least 57 genes related to redox biology, and all of them, nobody have studied them. Also, I want to study the mitochondrial function and the oxidative stress. So now moving to my second aim, that is to determine the role of host nucleotidases in enteric apicomplexa infections. So I want to study that because, as I mentioned at the beginning, parasites cannot produce their own purines. So this is a required to study host nucleotidases is required for parasite replication of the host nucleotidases are important for parasite replication. Also are target drugs, and there are many unknown mechanisms in this pathway. So I study, so this is a normal picture of the invasion process on Toxoplasma. So as we can see here, toxoplasma kiss the host cell and secrete a bunch of proteins, so I call this kiss and spit. And then create this parasitophos back wall where the parasite grow and replicate inside the host cell. So at this point I was wondering if maybe this single kiss can change the metabolism of the host cell. So in order to study that, I use an inhibitor of actin inhibitor called cytocalaxin D that allow the kissing and spit but prevent invasion. So using this technique, I did a metabolomics analysis to see if the kiss and spit change the metabolomics of the host cell. So I studied this during 12 hours post-infection, and I found significant difference at nucleotide levels, glycolysis level, pentose phosphate pathway levels, and amino acid level. So I confirmed this result with glucose tracking. And also here I want to compare the kiss and spit metabolomics with our previous published full infection metabolomics. And we found in common these 9 metabolites. Now, here I want to highlight 2,3-bisphosphoglycerate and these purines. So 2,3-bisphosphoglycerate is an allosteric activator of one enzyme called cytosolic nucleotidase 2. And this enzyme participates in the production of purines. So let me explain this hypothesis. So I believe that the kiss and spit of Toxoplasma gondii increase the abundance of 2,3-bisphosphoglycerate. And this one activates the cytosolic nucleotidase 2 enzyme that phosphorylate GMP to produce guanosine. And this guanosine is taken by the parasite to be able to create DNA and replicate. The same happens for IMP. IMP is phosphorylated by CN2 enzyme in order to produce inosine, and inosine is taken by the parasite. In order to test this hypothesis, I use a drug called fluorodavine that inhibits CN2. I test at 3 different concentrations, and the drug inhibits the replication of the parasite over time. So then I run metabolomics in these 4 different conditions in the presence of fluorodavine and without fluorodavine. And what we found was, I will focus here in the purple condition. We found that the products, GMP and IMP, was increased, and the substrates, GMP and IMP, were high in abundance, and the guanosine and the inosine, that are the products of this reaction, were downregulated. So it looks like the CN2 also is affecting, we confirmed that the CN2 is affecting the production of these purines. So also other technique that I use is click chemistry to evaluate the inosine incorporation. So we evaluate the inosine incorporation in 4 different conditions, and we found there is more inosine incorporation in the presence of the CN2 knockout hot cell line. So there are many questions to answer because this is a very complex pathway, how the parasite take purine from the host cells. So I want to continue this study using click chemistry, nucleotide metabolic profile, and intestinal metabolic cartography of spatial cartography, and gene expression and knockout of CN2 in other different cell lines. Finally, my aim 3, that is to study how Cryptosporidium parvus manipulate the host to meet parasitic glycolytic demands. Again, it's a known mechanisms, are target for drugs development, and is key to understand this colon cancer development. So I did some preliminary experiments infecting mice and treating with STAC, that is a protein licensed from Toxoplasma gondii, and STAC reduced the infection of Cryptosporidium. So then I moved to do a gene expression of the cocoon and ileum of these infected and infected mice, and we found that this solution STAC activate the interferon type 2 response. But in addition to that, what I want to highlight here is that the STAC also produce downregulation of 3 important metabolites, these 3, that I want to continue studying in my future lab. So how I'm going to finish or to continue this aim, so I want to do some target metabolomics in cryptosporidium also to study the glucose flux in infected cells, do some spatial cartography, and complement with gene expression analysis. So with that, I just want to say thank you to my two mentors, Dr. Melissa Scala and Laura Knoll, and my collaborators in the SPEED project, and all my collaborators in my K99. To the leading edge, thank you so much. And here are pictures from my two labs, the Knoll lab, the parasitologist lab, and the Scala lab, the engineering lab. Thank you so much for your attention. Let me know if you have some questions.
I'm sorry, I could be better at catching. So I was curious to know a little bit more about STAG and how treatment with that changes the gene expression during infection, and like what that indicates about those genes that you're going to study in the future.
Okay, thank you. So STAG has been studied before for other infections. I think was tested before for malaria, Plasmodium bergei, and reduced infection was tested again influenza, and also bacterial infection, I think was Listeria. And in all cases, reduced infection, but they never run gene expression analysis. So we infect the mice with cryptosporidium, we collect the fecal samples, and we found and we treat with STAG, and we found reduction on the shedding of oocysts. And then we do the gene expression analysis, and first STAG stimulate interferon type 2 response. So I think this is the main mechanism why it's reducing the burden of the parasite. And second, also, we found that downregulates this gene, lactase and glucose 6-phosphate and the fructose bisphosphate. So at this point, I don't know the answer, but I'm very curious to continue this project. But I don't know how STAG is downregulated this gene. We don't know. But I think it's very important, especially because lactase, so there is some preliminary data that say that people who is intolerant to, so some people become intolerant to lactase after cryptosporidium infection. There is some preliminary data. But I want to follow up on this preliminary data and continue with that, but I don't have the complete answer yet. Thank you, Teresa.
So I had a question. I thought it was really interesting how your infected cells, they became, they had an increase in oxidation. And so I guess my question is two parts. One, is this something that typically happens to cells post-infection of any parasite? Is it specific to your parasite? And the cell becoming elevation and oxidation, is that actually help the parasite, or does that actually create a condition that potentially prevents it or, from becoming more virulent?
I think it's like somebody here was presenting like it's like a battle, it's like a fight between the host and the parasite. So both have a antioxidant response, but at this point, I think we don't know what is the cause of that. We know that during infection, there is a regulation in the parasite of a lot of genes or transcripts related to redox biology, but also the host response. So it's like a balance between both conditions. And yes, the other question that if it's common in other parasites, yes. Using similar technique like FLIN of optical metabolic imaging, I have found that other bacterial infection, nobody have studied in parasite before, but other bacterial infections show also more oxidized environment over time.
Interesting. And then I guess it is a related question. I was curious, it seemed like you had a pretty global decrease in metabolism in your mice.
Mm-hmm.
I guess I just thought that was kind of interesting as to how it is so global. Does that make sense?
Yes.
You could speak to it?
Yes, in the cryptosporidium infection, how these mice were infected and also treated with STAG and then regulate many genes. Yeah. Yes, I have never thought about it. I don't know.
It just seemed a lot wider than I don't know.
Yes.
But I'm also not that familiar with the field, so.
Yes, and also it's interesting because stag came from toxoplasma, so we are using toxoplasma to treat cryptosporidiosis. So yes, but this is what I want to continue studying. I don't know.
No, it seems really interesting. It seems very, very interesting. Any questions? Yeah.
Hi, that was a really nice talk. I have a question. I'm not sure. It's very much not my field. But how does your host cell deal with overproduction of various nucleotides without the infection subsequently?
So
Is there any regulatory effect?
So how the host cell regulate the nucleotidases is the question?
Yeah, because you triggered this, but then you didn't let the infection happen. And so are you also seeing anything that the host cell is just
yes, that's a very interesting question. So far, we know that the single kiss and speed that doesn't have full infection, so the parasite cannot go inside, change many things in the host cell. So we believe is these proteins that I present at the beginning, like the rhabtrias, the micronins, and the ferrical bodies proteins that are secreting during this kiss and speed, injected inside the host cell, and these proteins are the ones who are changing the metabolism of the host cell. It's not a full infection. And also because we try by optical metabolic imaging the kiss and speed, and we detect early changes, even at 1 hour or 15 minutes post-infection, we detect changes not only at the redox ratio, but also fluctuation in ADPH and FAD. So we believe it's all these proteins that are secreted during this kiss, or the combination of all of them. So my future project is to start checking to knockouts of these rhabtrias, to determine what is the most important of all these rhabtria proteins or spherical body proteins. But yes, so far we can say that a single kiss of the parasite can manipulate the metabolism of the host cell.
Can you prime the host cell so that if you do a conditional mutation, and then so the parasite can infect immediately, but then the cell has time to deal with what's happening, and then you wait half an hour or an hour, and then let the parasite infect? What do you think would happen?
I think we did some kind of similar with just the media. Just we have an infection, we take just the media without any parasite, and add to the host cell. So this media doesn't have any kiss and speed, no parasites, and didn't change the host cell, didn't affect the metabolism of the host cell. So for that reason, we believe actually it's the kiss and speed that is changing the metabolism of the host cell. But I want to follow up with knockouts to see specifically what is that is changing. Did I answer your question?
Thank you so much.
Yeah.
Okay, thank you.
Oh, sorry. Oh, where should we? Thank you so much. Thank you. And so I'd like to move on to our next speaker, Abby Cheruiyot. She received her PhD at Washington University in St. Louis and is currently a postdoc at Harvard University.
Thank you so much for inviting me. I'm really happy to share my work here at the Leading Edge Symposium. So most of my career, I've really been interested in the control and fidelity of gene expression. So we know that central dogma posits that our genes within our DNA are transcribed to make RNA and then translated to make protein, which does most of the biochemical work that creates life. But we know this is not an error-free process, and there are a lot of mechanisms that are involved in improving the fidelity of gene expression. So one such mechanism is RNA surveillance. So in my PhD work, I actually worked on a nonsense-mediated RNA decay, developing kind of what is important in mammalian systems for this RNA surveillance mechanism. And most of RNA surveillance really occurred during mRNA translations. That's when I really got interested in translation, and this is what I'm working on right now in my postdoc work. I became really interested particularly in the role of the ribosome itself in the regulation of translation, because for a while we've thought of the ribosome as a constitutive translator of any mRNA that's poised to be translated. but there are new findings in the field suggesting that the ribosome itself can be heterogeneous and regulate translation of specific mRNAs. My work right now is applied to a very challenging public health problem globally, really, which is diabetes. And I'm really happy that Kristen has really introduced diabetes to all of you. And the global view really is that, so although the incidence right now in North America is really high and it's going to be higher, you can look at the subcontinent here in Africa that it's projected to increase by 134%. So this is becoming and will be a very global problem. So I particularly work in the pancreatic beta cells, which are these highly specialized cells within the islets of the Langerhans, which has other cell types. And the islets of Langerhans make up 2 to 3% of the pancreas. So this is very limiting material, which will be relevant later for my work. But it's a very good model to study translation because it has a very highly developed ER. The insulin mRNA actually makes up 30% of the mRNA, and the half-life is long. And the insulin protein makes up 10 to 15% of the proteome. And of course, I'm studying this because it's particularly relevant to diabetes development. So the reduced cell number of these pancreatic beta cells, their dysfunction or compromised identity leads to the development of diabetes. At the biochemical level, insulin secretion is actually tightly coupled to glucose metabolism, which makes sense. So if the cue for insulin secretion is glucose exposure, such as after having a meal, then there is a mechanism for this to work. So the glucose is taken up by glucose transporters on the membranes, which is then used in glycolysis and oxidative phosphorylation to make ATP. And it's the increase to ATP to ADP ratio that leads to closure of the KATP channels, opening of the calcium channels, influx of calcium that then supports secretion of the insulin. And we've known for a while now that this is highly supported by upregulation of insulin translation. So while the nascent mRNA remains the same, when beta cells are challenged with high glucose, insulin protein synthesis goes up about tenfold, and this is within minutes. So this is on the x-axis here, you have minutes. So this is a very specific upregulation of insulin protein synthesis. And now more studies have shown that it's not just insulin, but other genes involved in the insulin processing and production are also upregulated similarly at the mRNA translation levels, which suggests that there's a very coordinated mRNA translation upregulation for these genes for beta cells to do its function. And that was really something that got me really interested, especially interested in translation. So what about in the chronic or the pathophysiological state? So in diabetes, you have chronic hyperglycemia. Kristen described that. And so this was an experiment in human islets to really model hyperglycemia ex vivo. And they showed that if you do a 7-day incubation with low glucose, which is close to physiological range, or high glucose, these islets, as expected, when you challenge them with stimulatory glucose, they secrete a lot of insulin. But if islets are cultured in high glucose, they already produce or secrete more insulin even when they are being put in a basal glucose condition. And then when you further stimulate with glucose, they no longer release more insulin. So there's a defective mechanism of glucose-stimulated insulin secretion. Interestingly, this study also showed that the translation that was predicted to be increased by glucose is abolished in the high glucose condition. At this point, we didn't know if the other genes, the translation of other genes are being affected. So when I joined the lab, one thing that I wanted to look at is how does chronic glucose exposure affect genome-wide translation programs in beta cells? Because I believe there's a coordinated mechanism of doing this, and it might be affected by chronic hyperglycemia, which would be relevant to disease. So in the next 9 minutes, I'll tell you a little bit about the work I've done using different methods to measure translation, validation in different models, and what I think, moving on, what the mechanisms are. going to be. So I'm using a cell line model to do this high throughput experiments. It's been 6 cells, and the model I'm using here for hypoglycemia is a 24-hour treatment with high glucose and then performing glucose-stimulated insulin secretion. Again, here it's just taking the supernatant, insulin supernatant after basal glucose exposure and after stimulatory glucose exposure. And as expected, for the cells incubated in the low glucose, they are able to secrete a high amount of insulin when challenged with high glucose, but they lose that when you incubate them in high glucose conditions. So this was a model that I then used to check a few things. So first I looked at insulin translation specifically, and this was decreased, although total translation was actually not affected. And the ERs we didn't detect any ER stress resulting from the high glucose exposure. So I then used ribosome profiling to look at genome-wide translation efficiency. So in this method, I use cyclohexamide to essentially freeze the ribosomes on the mRNA and then generate 2 libraries. So one is an RNA-seq library that just gives me a snapshot of the abundance of the mRNA. And one is a ribosome-protected footprints, which I generate from using nucleases to chew up all the mRNA into space by the ribosomes, leaving me with these short fragments. And the assumption here is the more the fragments, so the more the ribosomes on the mRNA, the more efficient the mRNA is being translated. So in the end, I have an RNA and RPF and translation efficiency, which is a ratio of RPF to RNA. So glucose has effects across the board. So on the x-axis here is just a fold change, and on the y-axis is negative log of FDR. So on the left, you have genes that are downregulated by high glucose, and on the right, you have genes that are upregulated by high glucose. And I'm really most interested in genes for which translation efficiency changes, because I'm looking at the regulation at the translation level. And for this presentation, I'll focus on things that were downregulated by translation efficiency by high glucose. And this was nice to see, to validate previous studies, that this was included the insulin processing pathway, including the insulin genes themselves, ER to Golgi transport, glucose metabolism, and these key pathways that are important for beta cell function. But I wanted to use another method to look at this translation, because while ribosome profiling gives me a measure of what was bound by the ribosome versus what was free, it doesn't measure actual peptides. So sometimes you get a false positive due to translation pausing, for example. So I wanted to measure the peptides themselves. So I use a methionine analog to label all nascent peptides. And then this is clickable, so I clicked that to biotin and used streptavidin to immunoprecipitate all the nascent peptides and perform mass spec. So here I'm showing you results from this nascent proteomics, and glucose has effects across for many genes, but I was really interested in the overlap between the ribosome profiling and the translation and the nascent proteomics. And this included key genes important for beta cell function, including genes that function in glucose metabolism, insulin, and insulin secretion. So now I'm going to show you the validations that I've done. So the first is doing this ex vivo. So I did my discovery using a cell line, but I wanted to go back to now extract islets from rats because they're bigger, have more sample compared to mice. And then incubate that for 4 days in high or low glucose, and then perform validation by looking at ribosome-associated mRNA versus total mRNA. And as you can see, for all the genes that I selected to validate this, indeed decrease by translation efficiency by high glucose compared to some controls that I picked here. I looked at protein. It's also decreased at the steady state level for protein. And I don't have time to show you human islet data, but this is also phenocopied in human islets that I got cadaveric donors to test effect of high glucose. But I wanted to show you in vivo work too. So you could argue that what we are measuring is something that's artificial ex vivo. So I wanted to do an in vivo model of hyperglycemia. So here we did a partial pancreatectomy model of hyperglycemia. So we're essentially taking out about 90% of the pancreas, which causes artificial hyperglycemia because you don't have sufficient beta cells. And we wanted to choose this as opposed to diet-induced hyperglycemia because we can separate the effect of insulin, which is usually common in the diet-induced. Usually there's some compensation and increased production of insulin. So we wanted to separate effect of insulin versus effect of glucose. So here I was able to do the same validation using ribosome-associated mRNA versus total mRNA. And here I was also limited. So after performing the pancreatectomy, the amount of samples I was able to get, I was able to validate the most abundant genes. So this includes insulin and a few other genes involved in insulin processing and formation of secretory granules. But so far it holds true in vivo. And I looked at protein abundance. This is also decreased by high glucose. So I hope I've shown you that hyperglycemia is affecting translation efficiency of key genes at almost every step of glucose-stimulated insulin secretion. So from the transporters, metabolism genes, the insulin itself, things involved in insulin processing. So what are the potential mechanisms? So right now, because we think it's a very specific translation, so it's not a global change in translation, so there must be features within the mRNA that promote this. So I'm testing the UTRs of these mRNAs, looking at their contribution to translation. I've also done ribosome proteomics of actively translating ribosomes, and I've actually found some evidence that there's remodeling of the ribosome itself. So the compositions are slightly different, and we're very excited to explore what that role, what this heterogeneity has on the translation of these specific genes. And in my future work, one thing that I did find during my studies, I pressed something. One thing that I did find when I was looking at my data, some of the genes that function in the RNA surveillance pathway that I studied in for my PhD, specifically the nonsense-mediated RNA decay, was acutely downregulated by high glucose. And there's this now finding in the field that there's ribosomal products that contribute to T-cell attack of beta cells in type 1 diabetes. And these are usually new products. They are not your regular peptides that are from complete genes. So they could be resulting from alternatively spliced products or things that were not degraded by RNA surveillance. They could also come from errors from the ribosome itself. So my thinking is, moving forward, I'm really interested in exploring the role of remodeled ribosome, if we can find solid evidence that this is being affected by hyperglycemia, and also the role of RNA surveillance. So if it's being suppressed in the context of hyperglycemia, could this contribute to the production of these new peptides that are then secreted by the beta cells that contribute to beta cell attack? And there's actually a new paper that just came out a few days ago that suggested that in the context of type 1 diabetes, the observation is that they're seeing hyperglycemia even before appearance of the autoantibodies, which raises the possibility that hyperglycemia itself could be contributing to the immune attack of the beta cells. So I'm really excited about that. And I would like to thank the Schaefer Lab. So keep doing this. So this is my mentor. She's been very supportive. I'm really happy. And I would like to thank our collaborators, my funding sources, and all of you for listening, Leading Edge. And yeah, I'm glad I finished on time.
I'll hand it to you.
Oh, yeah. Sorry. I've lost my catching privileges. So I was really curious, when you think about how long to put them in glucose to model like chronic hyperglycemia, how do you think about that?
So the test for us to determine whether this is a chronic hyperglycemia is, what does it have on the glucose-stimulated insulin secretion? So if that's impaired, then it's a signal that it is already affecting the normal glucose-stimulated insulin secretion. And ideally, especially for primary islets, we want to make sure that we get the earliest time point possible, because the longer we culture these cells, the more they de-differentiate and they lose their identity. So.
This is a great gene list that you have. I mean, obviously, expressing insulin might rescue this, but I wonder if expressing any of these other genes that are downregulated is sufficient to rescue the insulin sensitivity of the cells.
Yes. I think it's a coordinate. So you need both. You need to make insulin. Actually, there have been studies that suggest sometimes the defect is not the insulin content itself. The defect is the release. So that might be actually another key thing to look into if you cannot make more insulin. Because actually, what's been fascinating in the field is that insulin content, there's just a lot of it. And during glucose-stimulated insulin secretion, they secrete about 2 to 3% of the content. What's fascinating is other studies have shown that the proportion of newly made insulin is higher for the insulin that's released. So we don't really understand why that's the case. We don't know why do they keep stocking up all this insulin if they're not releasing it. So there's still some biology there to be understood for is there any defects in the old insulin granules versus new insulin granules. But I do think sometimes part of the problem is the release. Yeah.
Hi, Abby. Great talk. Megan, Colorado again. So question, like thinking about the different types of beta cells within islets and the different stages, like does your data with chronic glucose exposure like point to any signatures amongst those 4 different types?
Yeah, so we haven't gone deeper into heterogeneity of the beta cell, and we hope we can do more with the single cell analysis of that, because this was like kind of like the quick and dirty, everything together. But we can do, and it's feasible to do single cell analysis of these now. With the translation, it's a little bit challenging, but I think we can get there. There's a paper that I've seen of single cell translation changes. So I think we could get there.
Okay, and a follow-up question to that would be more so speaking towards the de-differentiation.
Yes.
So as you alluded to earlier, like chronic exposure to high glucose levels leads to the selfish beta cell where it will de-differentiate in order to protect the cell. So have you seen, or have you allowed it to, I know you said you're trying to prevent it, but have you allowed some to go on just to see that signature in terms of translation?
So we haven't. So for the data that I've shown you, we've checked beta cell markers and made sure that they're still beta cells. They still express the beta cell genes and don't express the unallowed. But I do think that's an interesting thing, because we could select for things that do remain, like, and see what's different about those. I like that idea. Yeah. Can I?
Well, if there's no more questions, thank you so much, Dr. Cheruiyot.